Everyone, welcome to the Your Amigos Go to Hollywood video series. We are here in Southern California, actually in Monty Paul's backyard, which is quite nice.
Very nice, Monty.
Very nice.
Very nice.
We're here with Rana, McKay, and Monty Paul, and Tom and I. We're going to talk about papillary renal cancer, non-clear cell in general, but we're going to focus on papillary. So, Rana, why don't we start with you? Let's go back before the IO era. Papillary kidney cancer and the TKI era, and we're using sunitinib, and there were some studies. Tell us about basically activity of TKIs in papillary kidney cancer.
So a lot of the data that we have on rests on basically small randomized phase 2 trials that were conducted during the TKI era. The ASPEN study, the ESPN study that basically looked at sunitinib compared to everolimus and really kind of put a stake in the ground for sunitinib as sort of being a go-to standard of care for patients with non-clear cell RCC. Obviously no phase 3 data to guide what to do. There clearly is activity with single agent VEGF TKI, so that kind of set the landscape.
So what I would always tell patients is there's some activity, but it's not as much as clear cell, but we don't know what else to do. Right. And they were both fairly minimally active. And then, so Monty, talk about PAPMET. You were instrumental in getting PAPMET off the ground. Maybe just take us through what the vision was at the beginning and then what it ended up being.
Oh, sure, absolutely. So, it was just based on the general premise that MET has a real role in papillary kidney cancer.
I might challenge you on that, but go ahead.
Sure, we'll be happy to chat about that. And with that in mind, we looked at sunitinib as the control arm, given that all the trials that Rana had mentioned really served as the basis for establishing that. And we looked at several MET inhibitors. Now, initially, the trial was designed as sunitinib versus cabozantinib, very straightforward, 2-arm study. We took it to colleagues and other cooperative groups, and it expanded out to a 7-arm study. And in the way that cooperative groups go, it sort of whittled down ultimately to a 4-arm trial comparing cabozantinib, crizotinib, savolitinib to sunitinib.
And the other arms that never made it were?
Crizotinib and savolitinib. Those two
Okay.
drugs ultimately fizzled out at the time of a futility analysis.
Okay. And so talk maybe first about the arms that didn't make it. Not that were in the 4, but ultimately were closed.
Yeah. So, we always think of crizotinib as being an ALK inhibitor, right, in the context of non-small cell lung cancer and that 4% of patients that have ALK alterations. But it actually has some potent MET inhibition. There was some preliminary data from an EORTC study that seemed to substantiate its use in papillary kidney cancer. Not really a huge level of evidence, to be totally honest, ahead of the trial. But nonetheless, we didn't want to leave anything left to chance, and we wanted to make sure we explored all MET inhibitors, or to the greatest extent possible in our study design.
Are you sure MET inhibition is active in kidney cancer? In papillary kidney cancer?
There's some suggestion that it is, and I'll tell you, when you look at the data for savolitinib, and I'd probably turn to Tom, he's got a lot more experience with this, but I remember doing, the phase 2 study, it really does seem to suggest that in that population of patients that has MET-driven disease, and it varies how we define this. It could be MET mutation, MET copy number alterations and what have you. Seems as though the activity really stands out there. So in the MET-altered subset, I think there's probably a role for MET inhibitors. One limitation in PAPMET, is that we don't quite have that data yet.
Have you done that biomarker work yet?
Not yet, Tom, not yet.
And so the cabozantinib arm outperformed the other arms, but the sunitinib response rate was only 4%. Was there something wrong with the sunitinib arm in that trial? I think the cabo was 25% response rate, PFS of 9 months. Sunitinib 4 months PFS with response rate of 4%. Is that really representative? I mean, I use sunitinib in papillary renal cancer, and I think that feels low to me.
One of the big challenges in designing PAPMET is that the estimates for sunitinib at the time were all over the place. We had single-arm studies of sunitinib, like one that Nazar had run actually at MD Anderson, that showed a 0% response rate with sunitinib. So, we weren't really sure what to make of some of these varied data sets. Ultimately, we powered the study based on the SUPAP trial. And if I remember that correctly, the response rate was 10-ish% or thereabouts. 5-6 month PFS, which is ultimately borne out in the PAPMET study.
You only had 50 patients in each arm of the trial. It wasn't that many. And actually, when you look at the pembrolizumab data, single arm, they've got 120 patients in that trial. So although yours was randomized, it was a pretty small study. You had a PFS advantage for cabozantinib, but you never had a survival advantage. And the survival, even of the crizotinib and savolitinib arm, looked very similar. When you look back at that, do you think you can definitely say that Cabo's the best single agent drug in this, or do you think at the moment there's uncertainty?
I do think that based on level of evidence currently, Cabo probably should be considered the standard in this setting. Having said that, if you look at EAU guidelines, if you look at NCCN guidelines, it still seems to have kind of equal footing with sunitinib. So to be honest with you, I think if we showed a survival advantage, it would have been a slam dunk, right? I don't think anybody would have questioned use of cabozantinib in that setting. But given that we just see this modest PFS advantage, I think there's room to still explore a sunitinib control arm.
I mean, I think it is probably more active. I agree. Sunitinib in that arm probably was on the lower end of what you might expect, but so what? I mean, I don't know that it's that huge of a question.
And I think the PFS estimates actually jive with what we saw in single-arm phase 2 studies prior to that. 5.5 months is what we saw in SUPAP and other similar experiences.
So let's take...
Brian, we had a furious row on the way over here because you feel strongly that VEGF TKI IO therapy, the combination, and there are 4 or 5 different combinations, are your preferred option.
So when I look at the combo data, there's been 4 separate trials, right? There's been Cabo-Nivo, Len-Pembro, Cabo-Atezo, and Axitinib? No.
Bevitezo.
Bevitezo, thank you. How could I forget? Bevitezo. And they all have, pretty robust response rates, right? In the 40, 50-ish%, pretty good tumor shrinkage. So I know it's not randomized, right? There's never perfect data in non-clear cell, right? So I think IO-TKI is the standard of care for papillary renal cancer on the basis of higher response rates, more tumor shrinkage, and the fact that VEGF TKIs don't cure anybody. So if it's me, I want an immune agent in my regimen. Rana, what do you think?
Yeah, I actually agree with you. I feel like I really struggle when I have a patient before me that's got, non-clear cell RCC, particularly papillary RCC, of only giving them single agent VEGF inhibition or only giving them single agent cabozantinib. With the data that we have in the clear cell, studies, also the data that, building on the work by Jolie and others, sort of the combination strategies demonstrating much higher response rates, I really struggle with holding back. And I think that getting it right with the first line regimen is critically important. And so we already have data of the single agent IO data from the PEM study, I think 427. We have the single agent data, and now we have the combination data. And while we're not really comparing across trials, we do see a higher signal of activity with combinations.
I mean, Keynote 427 showed single agent immune therapy is active in papillary renal cancer, right? Somewhat impressively so. So it just kind of makes sense to me.
Although its PFS was short and its response rate wasn't spectacular.
And there was a high rate of early progression, so it's not
biomarker
perfect.
didn't work that well.
It's sort of like classic immune therapy where it works really well for a subset and not so well.
But is there a halo effect to that? Were you looking at that, and is it a case with the emperor's new clothes, Brian? So let's talk about VEGF-targeted therapy. Papillary renal cancer is not driven by VEGF. So we're happy to say VEGF-targeted therapy is a standard. There's no randomized trials to support VEGF-targeted therapy. If the breast cancer doctors were using it, they'd be giving hormone therapy on the same rationale. So we haven't got that right, let's be honest about it. And the second question is immune therapy. We've gone in with single-arm trials, no proof that they work either. And we're going with that too. There's no biology that I can see behind it, very low TMB, no particular drivers, no specific biology supporting it. And now we're giving the 2 drugs, neither of which we've really proven work, in combination, and we accept that as the standard of care, and that's what our furious row was about.
But part of it is...
Tell me I'm wrong.
I would actually say I disagree with the no activity of VEGF inhibition in papillary. I
didn't say no activity. What I said is the rationale
Yeah.
for VEGF-targeted therapy is not there.
Yeah.
So I don't.
It's not a beautiful biologic story like clear cell, I'll give you that.
It's not.
Yeah.
But it's clear these combos are active.
Yeah.
And sunitinib has a 0% response rate. Monty said it a few minutes ago.
Well,
So I'm not completely wrong here. This is not a ridiculous.
So Monty, how do you treat a new papillary that comes in, no contraindications?
Yeah, so to be honest with you, I think if you can identify the tempo of the disease, and I think in many cases you can, right? You look back, if it's a patient who's had nephrectomy, their a long time has elapsed to when they develop METs and so forth, maybe there's an opportunity there to go with single agent TKI. So my premise is, and we have a couple of studies in this papillary space. We have a trial of XL092 Nivo versus sunitinib. We have SWOG 2200, which is a follow-up to PAPMET, which is Cabo plus minus Atezo. If I look at that patient and say, okay, I've got time here, and maybe I want to spare that patient the toxicity of doublet therapy, right? I'll potentially consider one of these randomizations, right?
I think single agent TKI on a trial as a control is perfectly fine.
Okay.
But how about outside of a trial?
I would still consider single agent therapy for a patient with what I presume to be indolent disease, only because I think we've been burned so often by these single, keep in mind, for instance, Cabo-Nivo, the data that we've seen to date published in JCO is actually a single institution experience, right, from MSK. right? So how often have we been burned by a single center experience, right, that doesn't pan out when we apply it to the real world?
Yeah, no, I hear you. We're never going to have big randomized data sets.
Let's say we get Cabo Ipi-Nivo, that comes out, and that's got a slightly better response rate. Are you going to jump on the triplet therapy on the same rationale?
Good.
You're jumping on triplet therapy before
No,
you know it,
I'm
we're on
not.
triplet. But it's the same rationale, Brian.
I think adding a single immune agent to a TKI adds very little toxicity. Cabo Ipi-Nivo we know is toxic, right? So I think it's just a different equation when you're talking about triplets.
Can I talk about Savolitinib before we start?
Let's wrap this up. Let's talk about Savolitinib. Monty mentioned a couple big randomized trials going on, and I think Savolitinib is the other large randomized trial in papillary that I'm aware of.
So there's some work on a series of different trials. There's Samita, that was a randomized phase 2, was originally a randomized phase 3, but it ended up in a 50-patient randomized phase 2 for MET-driven tumors for Savolitinib, which is a MET inhibitor, versus Sunitinib, and the OS hazard ratio was about 0.5. Now it's underpowered, and it's
In
never
50 patients.
Yeah, well, in the same number as but the same number as Cabo versus Sunitinib is not and MET-driven.
That was 50 per arm, but
Yeah, but nevertheless, it's we're not talking 1,500 patient studies versus 50 patients. Okay, nevertheless, none of us are giving that as a standard of care, but we then did do a trial on those MET-driven tumors of durvalumab plus Savolitinib, and we showed a 53% response rate in those
immune
MET-driven
therapy plus
tumors.
targeted therapy. That's a great idea.
Well,
That's a really
but we've
great
looked at the monotherapy, and so you'll be pleased to know there has been driven, that's again, but it's not approved, and we're not using it because it's experimental,
Right.
rather than standard of care. And we're now doing a randomized phase 3 3-arm trial of durvalumab plus Savolitinib versus durvalumab alone, looking at the component parts, with Sunitinib as the control arm in MET-driven tumors. Now, it's a huge undertaking to do a personalized study, but I think it is, for me, the biology is better driven, the trial design is more robust. I hope we can finish it. I don't know if we can. We need a lot of patience, obviously, but I think that question's a really important question. Until then, I think there's a lot of uncertainty in this space.
So let's wrap up this video. Papillary kidney cancer, we can say single agent TKIs have relatively minimal activity. There might be advantages to one versus the other, say Cabo versus Sunitinib, but none of us are wildly excited about single agent TKI. Immune therapy has activity by itself, as shown in Keynote 427. Some of us believe that combos are standard of care, although based on limited experience. And there are, I think, the most encouraging thing, we've mentioned 3 randomized trials in papillary kidney cancer, right? And the last time that happened was maybe never, right? So I think the field, credit to Monty and others, has done a much better job of investing in and investigating papillary kidney cancer. But we all wish there was better levels of evidence for these patients.
It's a tough thing to summarize. That was well done.
Thank you, Monty.
Nice work. Thanks.
Thanks for joining.